we're live now. Good morning, everyone. Welcome back to our seminar series on Translational Biomedical Engineering. It's a great pleasure for us to host Professor Dino Di Carlo from Department of Bioengineering Department at the University of California, Los Angeles, with us today. But before we start the talk, I would like to go over like our housekeeping note. As usual, if you have any questions from our speaker, please use the Ask a Question box, which is in the bottom of your screen. You can also share your thoughts with us and our speaker and with other participants using the chat box, which is on the right hand side of your screen. I'd like to introduce our next speaker, Professor Catherine Granfield, who will be coming to this event from McMaster University, and she will be talking about some of the cool technologies that she's developing in biomaterials and biointerfaces in this area. So our next speaker is sponsored by the Canadian Biomaterials Society. So if you would like to receive most up-to-date information about these e-seminar series, please make sure that you follow us on Twitter. Our handle is TransLBME. You can also contact me and Human via email, or follow us on LinkedIn and Twitter to again get more information about these talks, and also provide feedback to us. And last but not least, we would like to thank Montreal Transmed Tech Institute, who has been with us since almost the beginning of these series. This is our 84th talk. We started in June of 2020, and then Montreal Transmed Tech has been with us since then. Thank you, Montreal Transmed Tech, for all your support for this event. So with that, I'd like to introduce our speaker. Professor Dino Di Carlo is very famous in the field of microfluidics, but I'll try to summarize his accomplishment in a few sentences, because if I want to talk about his work and accomplishments, then it will take a few hours to finish. But Professor Dino Di Carlo is a professor and vice chair of graduate studies at the bioengineering department at UCLA. He's also affiliated with mechanical and aerospace engineering. His research is exploiting unique physics, macro environment control, and the potential of automation associated with miniaturized systems for applications in basic biology, medical diagnosis, and cellular engineering. He has received many awards, but a few of them are Presidential Early Career Award for Scientists and Engineers, Material Research Society Outstanding Young Investigator Award, NSF Career Award, DARPA Young Faculty Award, Analytical Chemistry Young Innovator Awards, and then the list goes on and on. So thank you very much, Professor Di Carlo, for accepting our invitation to give a talk at our seminar series. And without further ado, I'll let you start your presentation.
Thank you so much, Mohsen and Human, for the invitation to speak, and it's my pleasure to share what we've been up to most recently. And I'd like, as Mohsen indicated, we've been working in the microfluidics field for a long while and done a lot in cell separation and other inertial microfluidics and other areas. But I'd like to share some of the newest work that we're doing in an area we call lab-on-a-particle technology. And the idea here is, can we do microfluidics without the channels, basically, and leverage really the instruments that are in labs now and particle technology to get to some of the kind of key features of microfluidics, which is creating small confined volumes to perform assays and reactions and measure those reactions, but do that without the need for new chips, new instruments to run those chips. And so that, we hope, will democratize, will expand the ability for life science researchers, for clinicians to use these microfluidic technologies. So I'm going to discuss our approaches for lab on a particle technologies. And where I like to start is, why are we doing this? What are the application areas that we can use techniques to look at individual cells and individual molecules? And I think the place that I like to start is what I refer to as the 3 pillars of medicine as we go forward into the future. We are very familiar, obviously, with molecular therapies. These are small molecule drugs that have been kind of the bread and butter of the pharmaceutical industry. More recently, we have now 5 out of 10 of the blockbuster drugs are based off of biologics or monoclonal antibodies. And so that's definitely another important class that's growing exponentially. And cells, B cells are the ones that produce these monoclonal antibodies. Obviously, we've heard a lot about RNA, and that's now an emerging part of this molecular therapies that is having more and more impact. The second kind of column here is gene therapies, and we have had recent successes and FDA approvals of the first gene therapies, and we have, of course, huge innovations in the way in the precision in which we can engineer the genome using technologies like CRISPR-Cas9 and related targeted genome editing technologies. And of course, the approaches to deliver new genes into cells using viruses, lentivirus, et cetera, those have been a growing area. You can imagine that when we want to select, for example, cells that are producing viruses or understand libraries of knockouts at the single cell level, here's places where you want to look at individual cells and how they're functioning, and then sort and analyze what's happening at the individual cell level. And then the third area, which we've seen a huge amount of success more recently, especially in immunotherapies based off of T cells and chimeric antigen receptors, is in the cell therapy space. And so this is a place where we're interested in the function of cells and how they interact and kill other cells, how they secrete cytokines, how they might secrete other drugs, how bacteria might also produce compounds that are beneficial, how tissues behave. In all of these cases, analyzing the individual cells, what cytokines they're producing, how they're interacting with other cells is critical to advance the field. And so the idea is that for these 3 pillars of medicine, analysis at the single cell level, at the single molecule level, that's critical to expand the rate at which we can develop these new therapies and bring them to patients. So the idea here is that we want to automate these biological processes, and the traditional approaches in which we perform automation or in the vessels in which we do studies of cells, they haven't really matched the biology. So, bioreactors, obviously a much larger scale. You go down to like Petri dishes, multi-well plates, even, these 1536 well plates, which require very specialized robots to operate on. This is still thousands of times larger than the volumes of cells that we're interested in looking at. And so we, in order to be sensitive in detecting what's happening at these cells, what these cells are releasing, what molecules they're secreting, we need the volumes to be similar to the size of the cell, right? So that we can concentrate the signal. And so this is where some of the microfluidic technologies like droplets have tried to address this by encapsulating, or microwells is another good example, encapsulating cells into these small containers so that when they secrete or release RNA or when lysed, that all of that material stays concentrated in that container and can be analyzed at high concentration, right? So the challenge there that I've seen through my efforts in commercialization of microfluidic devices is that even though the chip looks quite simple, there's always a big instrument around the chip, and that big instrument around the chip is very specialized. And it is not present in labs already. So if you want to now expand this technology and make it available to many people, that's a huge barrier. There's a much longer curve for adoption, even if the technology is very helpful. So we've been focusing on another approach, kind of microfluidics 2.0 approach, which we call lab on a particle. in which we don't use channels to create the, to create droplets and create compartments, or we don't use microwells in surfaces to create compartments, but we actually create particles that act somewhat like microwells, that have cavities that allow you to capture cells, to confine reactions, confine things that are being secreted, and that these particles are now compatible with instruments that are already present in labs, instruments like flow cytometers, instruments like single cell sequencing, and then can be operated with using kind of the standard equipment that folks are using in the research lab, centrifuges, pipettes, et cetera. So that's the approach that we're taking, really to match the scale of the biology of the single cells and potentially single molecule analysis, but use it in a way that's very accessible and that uses equipment that's already in the labs so that it can be distributed widely. So let me just go into a little bit more detail. What are these particles? We call them nanovials. I don't know if you can see, this is a 3D printed version of one. Obviously not to scale. You can think of these as much smaller. The cavity here holds about 1 nanoliter. Is about the size of the individual cell that can fit in here. And so because they're so small, you can put millions of them in suspensions, right? And you can put them in wells or in tubes. And so you can have greater than 1 million of these less than 1 nanoliter containing nanovials. You can then perform reactions here, right? So the first thing you need to do is load cells into these cavities. And so what we can do is we can modify the surfaces of these particles so that they have functionality to capture cells of interest. So let me talk a little bit about, in my lab, we've done a lot of work also on using hydrogel particles in kind of regenerative medicine applications. And so we took a lot of that knowledge, that expertise in making biocompatible hydrogel particles and apply this to this application area, so that these hydrogel particles that we call as nanovials, they also have a lot of the functionality that allows cells to be in a very good condition as they're interacting with the hydrogels. And so, and also that they have a lot of functionality for adding new moieties onto the surface of the hydrogels to interact with the cells. So we can add things like gelatin, we can add antibodies to capture cells based off of interactions with surface markers, et cetera. These, once you have a cell attached, we can also add antibodies to capture molecules that they're secreting. So let's say this is a B cell secreting an antibody, we can have an anti-IgG that would capture the secreted molecule. We can exchange reagents just by wash steps. So let's say you want to add things that will dye the secretions, you can do that, and you wash away things that are unbound. We can also form droplets around where the particles template the droplet, and that gives us now a confined cavity that is even more separated from the environment. And we can break that emulsion and recover those nanovials and cells. And I think the critical part, though, is that this whole process, all of this is then compatible downstream with methods to do analysis and sorting that are already present in labs. So let's say now you have labeled some cells that have secretion that are getting labeled by a secondary antibody that binds the secretion. You can then sort out through the FACS system in your lab, whether it's a Sony sorter or a Faxaria or whatever, and then sort out those populations and then separate them from other populations of nanovials and cells that were not secreting or did not have a particular function. And then you can do downstream analysis on these populations, whether it's regrowing them and using them in some experiments, or performing molecular analysis, genetic analysis of those cell populations and why they had a particular function. So let me just briefly, one slide on like how we make these nanovials, just for those who are interested. The process is actually a microfluidic process. So we take a precursor of multi-arm PEG with photoinitiator, and then we have a second part of the precursor, which is dextran plus a dithiol crosslinker. In this case, there was RGD that were a peptide that we're adding to also add some functionality to the nanovials for adhesion through integrin interactions with RGD. We have these co-flow, they form a droplet. When there's a PEG and dextran droplet and you control the concentrations in a particular regime, that droplet will actually phase separate into a PEG-rich region, into a dextran-rich region. And so you create these kind of crescent-shaped in cross-section. Obviously, it's more like a bowl, as I showed here with this 3D printed particle shape from the phase separation. And then once you have this phase separated shape, we can cross-link that using UV light to kind of keep the shape that was phase separated, and then bring it into a water phase, and then remove the dextran, which didn't have any cross-linkable moieties on it. And so that gives you this empty sort of cavity with these hydrogel regions around the cavity. We can then create a suspension of particles, and we can make millions, tens of millions, hundreds of millions of these particles. We can make them of different sizes just by controlling the microfluidic device. So here I'm showing particles from outer diameters of about 35 microns up to about 105 microns. And that, different particles are suited for different types of applications. So for some of the immune cells that are smaller, we usually use the smaller particles. Those smaller particles are more compatible with more instruments. And so like the Fax Aria and Sony sorters, generally they need to be less than about 60 microns or so to be compatible with these instruments. Although the larger particles can be compatible with other types of commercial instruments like Union Biometrica's Biosorter or on-chip sort. Obviously, all of these are compatible with like microscopes, pipettes, liquid handlers, et cetera. And the larger size are suitable for some other types of experiments. For example, looking at producer cells, CHO cells that are producing biologics or other bioproducts. Also stem cells, we usually use, although we use all different sizes for mesenchymal stem cells. And then if you want to look at clusters of cells or more than one cell interacting, some of the larger sizes are more beneficial. So the process to perform an assay with these nanovials has 3 steps. The first step is a cell loading step, where we need to basically load the cells into the cavity of the nanovial. And as I mentioned, the hydrogel particles that we make, these nanovials, have a lot of chemical flexibility. And so we've done a lot of different types of functionalization of the nanovials in order to enable lots of different ways in which you want to bind cells. So an example is RGD, polyl-lysine. I don't show it here, but we also have an approach to embed gelatin into the surface here, locally actually in the cavity. And we can also add like moieties like biotin. Obviously that allows a lot of flexibility in streptavidin biotin linkages. Or adding antibodies on the surface that are specific, for example, to cell surface markers to enrich a very specific subpopulation of a cell in your first step. So we can load cells. The process for cell loading, we've used a few different approaches. One approach is that we can just put our nanovials into a well plate and then seed cells on top, and they'll settle into the wells, let them attach. The other process that we've used is actually just putting a bunch of nanovials in a tube and then pipetting in cells and mixing it all up. And then depending on the ratio, if you have a lot more nanovials than cells, you capture a very large fraction of your cells into nanovials. This is just showing some data on the loading and how for different cell types, different types of loading are preferred. So I'm just going to kind of briefly go over this, but some of the CHO cell types, like the CHO DB12 is more adherent. It adheres well to RGD, and so we get good loading into RGD particles. XB CHO cells, we found that adding PLL or fibronectin, this is a suspension-adapted CHO cell line. It would help to get better loading of these cells. The HEC293s, in this case, we've loaded them with different things, gelatin, for example, but also we biotinylated in this case, and we see loading on cells, on the nanovials, and then B cells where we add antibodies against surface markers like CD19, CD45, or a combination, and we can get good loading of B cells into the nanovials. So once we load cells into the nanovials, we need to do some type of assay to characterize the function of the cell, so that we can then sort out cells based on their functional properties. For these applications, as I mentioned, in whether it's developing monoclonal antibody therapies, whether it's characterizing cells that produce lots of viruses or extracellular vesicles, or whether it's characterizing T cells or other immune cells and the cytokines that they're producing and sorting out subpopulations. So the first workflow I'd like to share is a workflow where we're looking at the IgG, the production rate of IgG by a CHO cell that's producing an antibody, therapeutic antibody. And so the workflow kind of goes like this. You start off by seeding the nanovials and loading the CHO cells into the nanovials. You can then add an anti-IgG capture antibody onto the surface. We allow the cells, in this case, we encapsulate it in oil so that we could close off the nanovials and prevent crosstalk. This is especially useful if you have really highly secreting cells, right, that are prone to just produce so much that it would get into other nanovials. And then we allow the cells to incubate for a period of time, secrete. We capture the antibodies that they secrete onto the surface of the nanovial next to the cell that's secreting. We then break the emulsion, add in the water phase an antibody that detects the secreted IgG and a fluorescent anti-IgG. And then we can wash that and then go through the flow cytometer to sort out the populations of CHO cells that are producing more IgG. So you can kind of see some results for a spiking, starting condition for a spiking experiment where you have cells that we know are not producing IgG CHO cells, and then an antibody producing CHO cell. And you can see a very different signal on the nanovials, and you can sort out the population that is producing antibody. So we use this to enrich and regrow a high producing subpopulation. You might want to do this if you want to then use this as a production line for producing your monoclonal antibody, right? You want the cells that are producing the most of it so that there's efficiency in your production. So we started off, in running the full assay. This is kind of after all the labeling steps. You can see a few nanovials here with cells and secretions. If you then go through the flow cytometer, in this case, you see this upper right quadrant are the cells that had a high secretion signal, this dialyte 488, as well as were cells, so cell tracker deep red. And so then you can see even here, there's a huge variation in the secretion amount for each of these cells. So you can see that histogram is coming from this upper right quadrant. And then we sorted both all the cells here that are secreting, so this whole histogram, and then the top 20% or so. And when you sort all the cells, you can see variations in the secretion. When you sort the top 20%, you see some, you see a little bit higher signal. And so the key thing, though, is like when we do this sorting process and we now recover a new population, do they regrow and do they produce more antibody in the end, right? Is this selection based on function actually yield a new population of cells that behaves differently over a period of time? So what we did is we expanded these populations after sorting, and then we could quantify the amount of IgG that was produced in the expanded population. And so what you can see is actually there's quite a large increase, up to 50% increase in production when we look at the top 20% versus the pre-sort population, or even just sorting all the secretors. So we're basically, it looks like we're removing non-secreting subpopulations of this, the CHO cells, and we're enriching the populations that had the high levels of IgG production. So that's great. This is after 10 days or so of regrowth. So that's also a stable phenotype, it appears. One thing that we found is actually in this process of sorting on nanovials that we improve perhaps because the cells are protected in these cavities, we actually improve the viability of the cells post-sort. So if you take CHO cells in this case, again, and you just bring them into suspension and you sort them through a FACS system, what we found is we get about a little less than 60% viability of those free cells after the sort, right? The process of sorting there's fluid shear stresses, there's a droplet that's basically deflected and goes into a well. And so there's a lot of stresses that are occurring as the cells are in suspension and being exposed. When the cells are in the nanovials, we saw a much improved viability because of perhaps this protective effect of being in this carrier. And we could regrow the cells after it, as I mentioned. So this is exciting, especially you think about adherent cells that you would want to sort, as well as very sensitive cells like actually when we do this with mesenchymal stem cells, we get even lower viability for the free cells, and we still maintain about 80% viability when they're on the nanovials. So this is an exciting aspect of using a carrier for flow cytometry to improve the viability of these kind of more sensitive cell types. So the next application I want to share is in not just looking at the production of an antibody, but the production of an antibody that is specific to an antigen, right? And so this is a key process if you want to do discovery of new antibodies to new antigens. An example is, during the pandemic, we saw rapid work, mostly using actually some of the first work was using microfluidic technologies to isolate B cell populations that were secreting antibodies that bound to the spike protein, and also, kind of were neutralizing antibodies to spike protein. So this process is a process of screening lots and lots of cells, right? You need to look at many, many B cells and look at their secreted antibodies, and then identify which ones bind to spike protein in that case. And then also link that information to the sequence information of those antibodies so that you can reproduce that information and now produce a genetically engineered monoclonal antibody in a producer cell line. So the process here is similar to our previous process. We load cells, in this case B cells, onto the nanovials. We have an anti-IgG to capture the secreted antibodies. We can optionally encapsulate in oil. Many of the experiments, we actually didn't do that. And the reason is when they're rare population that's secreting, the crosstalk is less of an issue. But then we can capture the secreted IgG. Then we can come in with a fluorescent antigen and see how well it binds to the secreted IgG. And then we can sort out this cell that secreted antibody, that bound antigen. And then we can put that into one cell into a plate, into a well of a plate, and then perform RT-PCR to capture back the heavy and light chain information about the antibodies that are being secreted. We started by just testing this out with hybridoma cells. In this case, it's a hybridoma cell line secreting an antibody against hen egg lysozyme. And so we loaded the hybridomas on. We then had fluorescent HEL. And so this is just a highlight of what the images kind of look like. You can see, okay, when there's cells on here, you could see high levels of the secretion signal, and you could sort out those subpopulations. So we then did a spiking experiment. We had these high HEL5 hybridomas in a background of 9E10 hybridomas that were labeled and were not secreting. You can see the types of signals you get when there is a secreting hybridoma versus a non-secreting. And then we could sort out, from this pre-sort population, a very rare population, less than 4% high HEL5 hybridomas. We could then pass that through the flow cytometer, sort this fraction here that had the high signal for HEL 647. And we could sort out those subtract fractions, and then we could characterize post-sort that we had greater than 90% purity of the cells that we are expecting that we wanted, which were the ones that were secreting the anti-hen egg lysozyme antibodies. We could then go and lyse off the nanovials and then look at mRNA, reverse transcribe it, and do PCR to, in this case, showing the heavy chain being amplified from these cells. You can also regrow the cells after the sort. So in this case, it's showing a hybridoma that's been sorted, and then it's regrowth after 3 days. If you even, as the days go further beyond 3 days, the cells just start to come off the nanovials completely. So it seeds the entire well. We then looked at doing a assay not with hybridomas, but with an immunized mouse model. So this is how you, one of the kind of the key ways in which you develop new monoclonal antibodies in animal models. You immunize with an antigen of interest. You then look for plasma B cells. You look for other B cells that are going to have specific production of antibodies against the antigen. So here we're using ovalbumin as our antigen. We immunized mice. We saw that, okay, yes, these mice were producing antibodies against ovalbumin. We could capture those antibodies on our nanovials. And this is just dilutions of the serum from the mouse. And we could see, even at around 20,000 times dilution, we could get signal on our nanovials. So they had B cells that were producing, this anti-ovalbumin antibodies. And control serum, you don't see that. We then performed our assay on the nanovials. We loaded cells. You could see that there's some subpopulations of cells that were producing lots of antibody. We could sort out those subpopulations, and we could now enrich populations of cells. So you see cell type. You see a few different things in this enriched population. You see some cells that are secreting a lot. And then you see some cells that had some surface labeling or intracellular labeling. And so you get all these varieties. So you're expecting some of the B cells will have a B cell receptor on their surface that can also bind antigen. And so some of these are also expected, although we're more interested in, and this is harder to do, is isolate the cells that are secreting IgG at high levels, like plasma cells, but don't have surface expression of the BCR. Great. So this is, I think we've got to a point here where this is kind of now laying the foundation to do full kind of drug discovery workflows with nanovials, where you can isolate and enrich populations of plasma B cells, characterize their secretions based on antigen binding, and then sort using all equipment that's already in your lab, and then isolate the sequences of those sorted cells. So I want to move on to another application area, which is in T cells and looking at the functions of T cells. So, this is related mostly to the third pillar that I had discussed, which is in cell therapies, where we want really functional, whether it's chimeric antigen receptor, T cell therapies, natural killer cells, macrophages, or engineered CCRs for T cells. We want functional cells that when they engage with an antigen, they're producing cytokines. They're producing the effector molecules that mean that they are functionally engaging, not just binding with some affinity, but not leading to functional responses. So the assay for characterizing T cell production of cytokines is very similar to the assay that we use for B cells. We seed the nanovials, we capture T cells onto the nanovials. We then have an antibody against the cytokine that's being produced, or more than one cytokine that's being produced. We allow the cells to secrete. We capture the cytokine. We then add a reporter molecule to label the cytokine on the nanovial, and then we can sort out the T cells based on their cytokine production, and then go down to single cell sequencing or downstream regrowth. This is showing some data on just the dynamic range of 3 cytokines that we're focusing on. So we're focusing on IL-2, interleukin-2, TNF-alpha, and interferon gamma, some of the key cytokines that are connected to the effector function of T cells. So we could detect quite low, down to 1 to 10 nanograms per milliliter on the nanovials by flow. It was better than by microscopy. Flow is a very sensitive approach using photomultiplier tubes for fluorescence detection. And this was sufficient for us to look at single cell secretion. So I'm showing some data on, after really the optimization process how we can characterize T cells based off of their secreted cytokines. Here on the top is interferon gamma, and on the bottom is TNF-alpha. But the idea here is that we allow them to secrete. We can label those secretions on the cells, and then we can sort out high, medium, and low. And so here's the gates that we use for high, medium, and low. And you can see here beautiful signal. Here you see, in this case, we looked at live cells, so we had a viability stain as well. The nice thing about this platform is that it is using flow cytometry, so you have huge like spectral range for many flow cytometers. You have many lasers for excitation and many kind of filter sets, and so you can add a lot of different stains, whether it's surface markers or multiplexing of the secreted markers, and I'll show some more of that. But yeah, you can see here live stain, so live T cells, and then beautiful signal of the interferon gamma around the rim of the nanovials and in the nanovials. It appears more bright around the rim because you were looking from the top, right? And if you think about it from the top, there's this edge of the nanovial that has a lot of area and depth. And so that edge is where we're seeing a lot of the signal. And it's very similarly for these TNF alpha producing cells, beautiful signal associated with the cells. And as you go to low and medium secretors, you can pull out different subpopulations. We can also do multiplexed secretion-based sorting. So in this case, we did TNF alpha and interferon gamma. So TNF alpha is on this axis here and interferon gamma here. So here there's a population of high interferon gamma secretors, high TNF alpha secretors, and then a combination that are these polyfunctional T cells that are secreting both interferon gamma and TNF alpha. So this, in these experiments, we were activating T cells using PMA ionomycin, kind of a non-specific activation. But it's still, you can still see kind of this subpopulations that we can pick up from non-secretors to a large fraction that are just secreting interferon gamma to a smaller fraction that's secreting more than one, and a really small fraction that's just secreting TNF alpha. We then kind of multiplex these assays with surface markers. So we're interested in CD8 positive and CD4 positive T cells and how their secretion profiles may differ, right? So if we looking at CD8 positive T cells, do they have a higher amount of certain types of cytokines being produced than CD4 positive T cells? And can we sort out based on the combination of surface markers plus secreted? And so in this case, we looked at multiplexing both interferon gamma and TNF alpha or interferon gamma and IL-2 for CD4 positive and CD8 positive T cells. And so we could pull out populations based on this combination. And so what we found is generally the CD4 positive T cells have more of this kind of polyfunctional phenotype where they secrete, both, whether it's interferon gamma and TNF alpha or interferon gamma and IL-2. There's as expected, the CD8 positive T cells secrete very little IL-2. There's only a small fraction that secretes IL-2, while there's a much larger fraction of CD4 positive cells that secrete IL-2. And I guess maybe unexpected to me was that there's also a large fraction of cells that don't secrete cytokines upon activation as well. So this is exciting data. We can kind of really, in a really simplified format, get into details of the subpopulations of T cells. And of course, this is applicable to other types of immune cells as well, in a functional way, and still keep them viable, right? So the ways that you currently would look at cytokines and be able to sort cell populations based on cytokines, there's not many out there. You can look at intracellular cytokine staining, but you've already killed the cell at that point in order to get antibodies inside the cell. And it's not clear whether the cytokines inside the cell are eventually will be secreted. And you can also do like something like an ELISpot, but in an ELISpot, you're not able to sort these subpopulations, right? You're not able to look at surface markers combined with secretions generally. A final approach that some people use is like you can add a capture antibody onto the surface of the cell for the cytokine. So this is a product that Miltenyi has. But it turns out, that for some cytokines like TNF alpha, there's actually a surface bound form and a secreted form. And so those are unable to kind of tell the difference between those two forms. and there's a lot of crosstalk if you have a lot cells that are secreting a lot, because you have no cavity to confine the secretions when you just label the cells themselves. So we extended this work not just to looking at PMI-inomycin-based production of cytokines, but also to look at antigen-specific T cells. So in this case, we had a MHC monomer that we put onto the nanovial rather than anti-CD445 antibody. So that MHC monomer now the nanovial acts as kind of an artificial antigen presenting cell. We can first, enrich populations of T cells that have the cognate TCR to bind that peptide MHC. They will get enriched and loaded at higher concentrations onto the nanovials. And then what we found is that the presence of that peptide MHC on the nanovial actually was activating the cells to secrete cytokines. So in this case, we see interferon gamma production upon binding solely to nanovials that had the peptide MHC monomer, but not nanovials that had anti-CD45, right? So it's the interaction with the peptide MHC monomer that leads to the interferon gamma secretion. And so in this case, we looked at T cells transduced with a specific TCR, so there's lots of these T cells around, and we could see a lot of signal. But then we started to look at, is this an approach that we can enrich for TCRs that don't have maybe as high affinity as well? So this is a collaboration with Owen Whitty's lab, and we started to look at a number of different TCRs that they've previously investigated against the NY-ESO1 antigen. And there's the 1G4 was the one data I showed you in the previous slide. You can get a nice number of cells loaded onto the nanovials, and then they also secrete interferon gamma. 92 is another TCR that we use that they were unable to see really great binding to a dextramer, so that means the affinity is quite low of this TCR. But even in our assay, we were able to pick up these cells, enrich them, potentially due to the avidity interactions with the nanovial, and also get really high levels of interferon gamma production by even this low affinity TCR. So that's exciting because you can start to find new T cell receptors that may not be easily discovered by just looking at binding of dextramer to the T cells. It turns out that dextramer binding is also quite a noisy assay, so you get a lot of false positive signals. In our approach, we shouldn't get false positive signals at higher, and we have data now that shows this. We don't get false positive signals because we're also looking at the function, right? If they bind alone, that's not enough. If they bind and then secrete cytokines, then we know that's a functional engagement. So that's exciting, and we're able to kind of use this across a lot of different TCRs, and we're now expanding to do new TCR discovery for new therapeutic options. I mentioned for other cell types you can regrow them after on the nanovials, and this is just showing that you can regrow also T cells after interacting with our nanovials. In this case, the peptide MHC nanovials we trypsinized after the sort, and we could regrow populations, expand them, after enrichment based on functional activation. So we've been expanding to a lot of different applications with this nanovial platform. We're super excited by it. Basically, the huge range of biology that we've been missing when we think about secreted products that we haven't been able to study at single cell level as we would like, because we can't sort populations of cells based on what they secrete. At least it's not available to most people. There are 1,500 secreted proteins in the human genome, which is about 2/3 of the amount that are on the surface of a cell. So that means that we're kind of missing almost half of the proteins that we're interested that are part of our genome that have huge importance in immunology and in all biology, really, because we haven't had tools to characterize these secretions from single cells. And so this is where we're very excited, and we've been expanding to other areas beyond just secreted proteins. So in this case, we're looking at the production of extracellular vesicles from mesenchymal stem cells. As you know, mesenchymal stem cells have been used as therapeutic options for a number of diseases because they have some unique properties, whether it's kind of regenerative properties or immunosuppressive properties. And it's thought that a lot of these properties are related to products that they produce, whether they're secreted proteins like VEGF or IDO, or whether it's secreted extracellular vesicles. So what we've been doing is looking at the secreted products of mesenchymal stem cells, and this is just showing you our assay for characterizing extracellular vesicles produced by single mesenchymal stem cells. So how that assay works is we have a capture antibody for the EVs, the extracellular vesicles. It's anti-CD63. We then have a detection antibody, anti-CD9. And so we're labeling now the double positive CD63, CD9 vesicles that are EVs that are being produced. What we found for these particular cells that we're working with is that that was the largest population of EVs were double positive for CD9 and CD63. This is showing data from a Amnis image stream, just showing that, yes, these nanovials are also compatible with imaging flow cytometry. And so here you're seeing the MSCs in green, and then the EVs that they're being produced and stained around the nanovials in red. And so you can do really interesting things here, where you can characterize the production of EVs and also kind of gate out any staining that might be on the cells as well. We've also been doing sorts based on EV production. So this is showing you the low secreting population, cells that secrete low amount of EVs, as well as cells that secrete higher amounts of EVs. And we could sort those subpopulations and regrow the MSCs to see, are there functional differences in these cells? Do they act therapeutically in a different way when we use them in models of disease? So this is very exciting. We can also do this type of secretion-based sort or EV-based sort at the single cell level to get clonal populations that can grow up from the populations that we select that have different phenotypes. So yeah, very exciting approach that we're now applying, you can imagine, to see if we can enrich the best populations for a cell therapeutic. Or let's say that you want to use EVs as a part of your therapy itself. How do you pick the populations of cells that produce more EVs to make your manufacturing more efficient? So those are applications we think are going to be really interesting. Okay, so I think now I'm going to kind of conclude, that if you think about, from the beginning, I was talking about, the scale in which we do biology. We've been, the kind of the smallest scale in which most of biology is being done is this 1536 well plate format, which has volumes about 1 to 10 microliters. This is obviously much too large for individual cells, so you have to usually grow up colonies in order to do anything. What we've been doing is using these nanovials or dropicals, where we encapsulate nanovials. And those are thousands of times smaller volume, less than a nanoliter. So that means that the concentration of the secreted products that are produced is concentrated thousands of times higher. It's much easier to detect in a short time. We can look at thousands of times more compartments, hundreds of thousands of events with our flow cytometers that can process hundreds to thousands of events per second. And you can sort these, right? So that's something that's also difficult to do in the multi-well plate formats, right? So you have the capability to, once you've seen a functional property of a cell that you're interested in, you can sort that out and study it further, whether it's through genetic means, transcriptomics, epigenetic means, or regrowing those cells and seeing, do those, how are those phenotypes important for eventual function of those cells, whether it's in vivo studies or in vitro studies. So this is very exciting, and the fact that you can do all of this without new equipment, with the flow sorters that you have already in the labs that you're already working in, this is where we're extremely excited, and we want to share this technology with as many people as possible. And so that's one of the reasons we also started a company called Partilian Bioscience that is commercializing this technology, and actually is already selling some of the nanovials that have biotin on them, so that they're very flexible for researchers to use. So I'd like to acknowledge Joe Giodaretti, who's leading Partilian, but was my PhD student in the lab, who did a lot of the basic work to get to this nanovial technology, from fabricating these to running assays, and many of my students who are continuing to do really great work on applying the technology, this lab on a particle technology, to many, many, many new different types of assays. Also thank our collaborators, and thank you for your time, and I'd be happy to take some questions.
Thank you so much, Professor Di Carlo, for this fantastic talk. If you wish, you can stop sharing so it's more interactive. Yeah. So since our time is limited, I just right away go to the questions. I have a couple of questions myself, and then audience, I encourage the audience also to ask their question and put them in the ask a question box on the bottom of the screen. So this is really a cool technology, and especially for single cell sequencing, and as you mentioned. So in some of the images that you show in the beginning, in terms of capturing different cells, I'm just wondering how much is the yield of capturing the cells. I know that the biomolecules are there, but how much is the yield and the performance?
Yeah. So that's a great question. So you can tune that. So let's say you have a very rare population that you don't want to lose any of the cells. What we do in that case is we use a lot more nanovials than cells. So we might use 10 times as many nanovials as cells. And then what happens is you have a much higher probability of getting every cell into a nanovial. And our efficiencies at that, we are usually getting like 50 to 60% efficiency of cells that you put in get captured on nanovials. There are other cases where you don't care. You have too many cells. You have too many. You just want a high fraction of nanovials to have cells. In that case, we'll change the ratio. So we'll have more cells, maybe 1-to-1 or 0.6-to-1. And there we get, we will get a larger fraction of the nanovials have cells, maybe 10 to 20%, but then we'll lose many cells too. So we might have 15 to 20% yield. So it really depends on the application, if you have a rare population that you want.
Perfect. Thank you so much. And in terms of retrieving the cells, you said that you use trypsin. Is there any other way, like, to kind of retrieve the cells without trypsin?
Yeah, so the main way actually is we let the cells regrow out of the nanovials. So as you saw, like where the MSCs, they grow back onto a plate. They'll kind of crawl out of the nanovials. And then for the hybridoma cells, as they grow, they'll just fill into the suspension. So trypsin we use in some cases if we just want to remove the nanovials from that culture vessel, but in most cases we'll just let them regrow out of the nanovials.
Perfect. And there is a question from one of our audience. Ali is asking, what is the biggest challenge of these nanovials?
I think what's been so great is that it's relatively, so being in a microfluidics lab, we've done a lot of microfluidics, and I would say that there is a learning curve in microfluidics. Even you see a paper out there like, oh, they did this droplet generator and they did all these assays with it. But it turns out that's really hard to do, to even making things very stable and doing all these assays. Droplet assay, all that is very difficult. So what we found is that the nanovial systems, we can get new students, new researchers up and running. Could be 1 week they're ready to go and run assays. So we haven't found there to be I guess, like challenges, like we've seen with other systems where there's kind of a learning curve. I think the key thing is that we're using flow cytometers. So if you have access to flow cytometer, then it's perfect. And if you are familiar with flow cytometers, but if you're not familiar with flow cytometers, then there's a learning curve on using flow cytometers. I would say that's maybe the biggest challenge.
Thank you so much. There is another question from Ilham. She thanks you for your great talk. And she's wondering whether one can use the nanovials for two-cell interaction studies, for example, to study the interactions of single bacteria and single phagocytes in one nanovials. And this technique, if this technique follow the Poisson distribution.
Yep. Yeah, those are great questions. So, so I can tell you that we have a collaborator now who's really interested in this idea of bacteria interactions with cells. So what they're looking at is they want to look at, once a bacteria infects a cell, intracellular bacteria, how does that change the secretion production products that are produced from that cell, interferon gamma or other cytokines and things. And so there's no way to really study that now and sort out that population and look at what's different about it. So yeah, I think that definitely there's ways to do, whether it's single bacteria and single cell becomes more challenging, right? Because like you said, Poisson distribution becomes an issue because our system, generally when you load single cells, we follow Poisson distribution. We can beat it a little bit by controlling the cavity size. If we make this cavity size similar to the size of a cell, once one cell is bound, then the second cell can't get in. And so you can go a little bit better than Poisson. But let's say you added now a bacteria, that's going to follow some other Poisson distribution. So if you want 1 and 1, it would be a little bit more challenging. But that's the same for any technology, right? Droplet microfluidics or microwells, you also have this double Poisson issue. So we have, the way that you can solve some of that is you can sort, because the sorting is so fast, right? You can sort only the nanovials that have cells. Now you have only single Poisson issue, right, for single bacteria.
Perfect. Thank you so much. There is another question from Farnaz. Is it possible to use nanovials hydrogel as a substrate for variable electrochemical biosensors to detect the cell secretion biomarkers?
Yeah, I'm not sure. So let's see. So as a substrate for wearable electrochemical biosensors. So I think the approach is more suited to optical sensing because you don't have a connection, right, to any voltage sources or anything to control the electrochemistry. You could do enzymatic kind of reactions, just like ELISA or other things on the nanovial. The way to think about it is a well of a well plate. Whatever you could do in a well of a well plate, you could do on the nanovial. So it's probably less suited for electrochemical assays and more for optical assays.
Thank you so much. I will stop here. I pass to Mohsen to continue his questions. Thank you.
Well, thank you very much. So Dr. Di Carlo, I don't know how much time you have, we have with you, because as I can see, there are many other questions are being added to the question box, and then I also have tons of questions, which I don't know how much we can
maybe like 5 more minutes. Does that work?
5 more minutes. So
okay.
Like, to be fair, I mean, let's wait, ask the questions from the audience woman first, and then
okay.
and then if there's time, then I always can drop by Dr. Di Carlo's office and then ask my questions.
Sounds good.
Chris, yeah, so I'll continue. Yeah, Professor Moraes from McGill University is asking, this was fantastic to hear. Without giving away any proprietary plans, of course, what do you envision as being the next great step forward for lab-on-a-particle platform?
Yeah, I mean, I think and I'm always very open about our plans. If you read some of our review papers, you'll see the plans for the next 5 to 10 years. I think the key here is, can we apply this technology to select out cells by function to make better therapies, right? So, and to discover, in the end, cells work, they function in biology based off of the things that they do, not their genome necessarily, right? The genome is part of that, but in the end, you need to know how they are actually behaving. Are they killing other cells? Are they secreting molecules? Are they interacting with other cells and in different ways? So we see this as a platform that expands the single cell analysis field into functions. For the first time, using all the equipment that's already out there, right? So that we can look at cell killing assays at the single cell level. What's different about a T cell that kills another cell very rapidly versus one that doesn't? What is different about cells that produce more extracellular vesicles than those that do not? What's different about cells that release lots of virus versus cells that don't? So all these questions that we couldn't ask, and we couldn't answer, so we didn't even ask before, right? So those are the areas that it's opening up, all these new questions, and so we're excited. We're not going to be able to answer all those questions ourselves, and honestly, I'm interested in the questions, but I want to help other people answer those questions, and that's my goal. And so that's why I want to get these technologies out there for others to use.
Thank you so much, Mohsen. I know you have questions. Please go ahead.
Well, I'll ask just one question about the commercial, some of the commercial activities that you're doing. And as you mentioned, that there's a company, a startup that is pursuing commercialization of this technology. Can you comment on some of the challenges that your team has faced during the commercialization of this technology? And specifically, my question is about sterilization, because these are going to be used with cells and you want to avoid contaminations. Did you face any challenges regarding the sterilization of these particles?
Yeah, no, that hasn't been a challenge. I mean, so since we've started working on it in the lab setting, we've used ethanol-based sterilization processes, and because during the fabrication process, no cells are around, so we can use very harsh. And actually, the cross-link hydrogels are very, very robust to a lot of different solvents. So when we actually make them, we're exchanging through lots of different organic solvents. So the sterilization process, we've actually never had issues with contamination from the product itself. So that hasn't been an issue. Certainly, scaling production is always an issue. You want to be efficient in the production of these sorts of things so that you can get it to more people at good, at a reasonable cost. So that's a continued challenge, and we're working on how do we scale further the production of the particles.
Well, awesome. Thanks. I mean, you talked about the scalability. I know your team is doing some work in the area of improving the scalability of these microparticles. Well, I think in light of the time that we have, I think I would like to just thank our speaker, and again also thank the participants, and then remind everyone that our next speaker is Professor Catherine Granfield from McMaster University, who will be talking about multi-scale characterization strategies to probe osseointegration and biomineralization. So thank you again, everyone, and then I think we can conclude this session today.
Thank you so much. Take care, everyone.
Thank you so much.
Have a good day.
Take care. Bye-bye.